Hi everyone, my name's Tom Rollins. I'm the marketing manager here at FORMULATRIX. I'd like to take this opportunity to thank everyone very much for taking the time out of their busy schedules to be here today. I'm really excited for this application-focused live web event. So before we begin, I'd like to provide an overview of the agenda. So first we have Paul Ventura, our Director of Sales here at FORMULATRIX. He'll be briefly introducing solutions for positive displacement microfluidic reagent dispensing for high-throughput qPCR workflows. Then, Denise Cruz, the Senior Market Development Manager at Labcyte, will explain the benefits of acoustic dispensing for nucleic acid sample transfers. Then, Sripathi, our scientific speaker from the Wilmer Eye Institute at Johns Hopkins Medical Institute, will explain his research and how the Mantis and Echo have been integrated into the high-throughput qPCR workflow in his lab to promote efficient and reproducible high-content screening. At any time in the presentation, please feel free to enter your questions into the GoToMeeting or the GoToWebinar interface, and we'll answer all that we can at the end of the live web event. So with that, I will now introduce Paul Ventura, the Director of Sales at FORMULATRIX. Take it away, Paul.
Thanks, Tom. Thanks, everybody. So I thought I'd start with a brief introduction of who FORMULATRIX is, briefly describe the technology that we've developed around our reagent dispensers, and then ultimately show you where we fit in the workflow for common qPCR applications. FORMULATRIX, from birth, has been a crystallography company. So the definition of crystallography is, in basic form, is taking a series of reagents, combining them in various concentrations with the goal of promoting crystal growth. From that, we also develop a series of imaging systems in order to capture and study those individual crystals. What spawned from that was a microdiaphragm technology that led to a series of reagent dispensers that I'll spend a few moments on today. A separate and third product line that we were beginning to announce, and this is more of a teaser, but I'll ask you to stay tuned at a later webinar, but is a new high-throughput plate-based digital PCR system that we expect to launch later this year. So stay tuned for that one. But who is FORMULATRIX? So FORMULATRIX has been around since 2002. We're a privately owned company. This slide is a little bit outdated. We've got about 350 employees now, an installed base of over 1,200 instruments globally. And I wanted to point out just a quick smattering on the side of who our customers are, but really identify that we do have support in sales and service operations out of both the US, Europe, and Asia Pacific areas. So the focus of the next few minutes will really be to key in on two of our reagent dispensers, the Mantis on the top, the Tempest on the bottom. Some of the key features of our technology has been our really low dead volume. We tend to migrate towards precious reagent applications such as genomics, high-throughput screening, areas where people want to minimize waste associated with traditional liquid handlers and prehistoric reagent dispensers. So that's one of our main features. Our instruments use our own patented microdiaphragm technology, which I'll describe in just a short moment. But the goal is really, it's a non-contact dispense, ability to dispense into any volume, into any well. And we also use a positive displacement technology that I'll walk you through. But some of the keys there is we get around all liquid classes. So we're very amenable to working with things such as spry beads, organics, that most traditional reagent dispensers struggle with is an area that we excel. So how does the technology work? Hopefully the animation will come through. But everything is based off of vacuum and pressure. So what both of our instruments do is the first thing is put our microdiaphragm pumps under vacuum. And what happens in the animation is you'll see an inlet valve opens up when everything is under vacuum. Then our microdiaphragm, just going to pause that for a moment, opens up. And this creates a vacuum which basically isolates a very precise volume of reagent. And that's all under vacuum. And then what we simply do is turn the vacuum off, put on positive pressure, we close the diaphragm, and that generates a precise droplet volume that is ejected into your plate. On the right-hand side, you'll see a quick video of it happening live. So as we turn the vacuum on, the reagent goes in, we then punch it out, vacuum goes on, we pump and cycle until you get to your desired volume. So I'll show you that again. Here we go. So vacuum comes on, the reagent diaphragm fills, and then we dispense that droplet out. And we can do that about 8 times a second. So it's a very quick technology, but it gives you a lot of precision over specific volumes that we can dispense. So what do the chips look like? These are our Mantis chips. I'll talk about the Tempest in just a moment. But these are about the size of a quarter. The cost on each one of these chips is about $60, and they're completely reusable. We have wash protocols and everything. So it's designed to be not a consumable for the instrument, but it's designed to be low cost, where if you're in, let's say, a core setting, all of your researchers could have their own set of chips. What I did want to draw your attention to are these small diaphragms, because those are our micro diaphragms for each type of chip we offer. The left-hand one is a low-volume chip that has diaphragms that have 0.1 and 0.5 microliters. We also have a high-volume chip, which has volumes of 1 and 5 microliters. So if you remember the slide from just a moment ago, what happens is this chip gets put under vacuum. These two cylinders fill up with 5 microliters here, 1 microliter here. We then turn off the vacuum, put on positive pressure, and again, we eject droplets of 5 and 1 microliter out. Let's say you want 3 microliters. Well, we just dispense 3 single microliter droplets into your well. And that's what we do. And again, we've tested these chips for over 2 million cycles without issue. So they're inexpensive, and they're designed to be a non-consumable and completely washable. All right, so this is what the Mantis looks like. It's a little video, but before I hit play, I just wanted to point out a few things. Standard pipette tip here. So let's say a researcher gives you a very precious reagent. Let's say you have 100 microliters and you want to dispense 1 microliter per 96 wells. We would ask you simply to put it in a standard pipette tip and use that as your reagent reservoir. Therefore, you're not losing any reagents to tubing, as in this example to the left, but it allows you to reduce your dead volume and reclaim as much of the reagent as possible to minimize any waste. So let me hit play. There we go. So what the Mantis does, it looks really, really good moving these chips around, is really what it does. It's plug and play, meaning that all you need to do is plug the Mantis into the wall. Everything is calibrated within the instrument itself, so there is no liquid classes, there is no pressure or vacuum that any end user has to worry about. Essentially, it's just plug it into your PC, tell it what reagent you want dispensed in what well, and you're off and running. So it's a pretty simple little robot. Might be hard to see in this, but we try to emphasize the size of the instrument, so it's no bigger than a toaster. This is a standard microplate. Oops, sitting on the deck. Hang on, back up. Right there, it's a microplate, so if you can envision that in your head, you can get a very good understanding of how small the instrument is. So the Tempest is largely the big brother to the Mantis. So while the Mantis can dispense any volume into any well, it does it serially, meaning 1 well at a time until the next well. The Tempest has the ability to use up to 12 reagents, but you can also dispense everything in parallel. So to give you an example, this top configuration here is 96 individual chips. So if you wanted to do very high throughput, we can do 200 nanoliters in 3 seconds across a 96 well plate. So this instrument is largely accompanies the Mantis. So often a customer that has a Mantis will increase their throughput and then migrate towards a Tempest, which gives you stacker capability, again, much faster dispense speed, but you also have the same ability as the Mantis, meaning that any reagent can be dispensed into any well. So you have a lot of flexibility, but you also have a lot of throughput. And both of our platforms are integration friendly, so this unit can come with or without stackers, and you can feed it right up to a robotic platform. So again, the applications that we migrate towards are the ones that have the most expensive reagents, because ideally, if you can quarter your reaction or tenth your reaction, not only does it mean you can save costs, but also means you can run more samples. So we look at genomics applications, of course, NGS library preps, and that means everything from doing bead cleanup with spry bead dispensing, ethanol, straight to your Nextera XT kits. So we can handle all of that process. We also do well with cell-based assays. It's a very gentle dispense, so even though we do use vacuum and pressure, it's very low shear stress, and its cell viability is equivalent to doing handheld pipetting. And another application where we have about 1/3 of our units is assay development. So because the Mantis and Tempest can dispense in between 6 and 12 different reagents with any volume into any well. You can create a lot of design of experiments and really generate data to point you in the direction of how you want to run your large-scale production runs. So to give you an idea, just in a qPCR workflow, kind of the areas that we fit in. So we start at the beginning. You can use our instruments for extraction, straight up to master mix and primer additions, and every step along the way. So it's not just used for primarily expensive reagents. It can be used for bulk dispensing media, or again, your ethanol for your spry bead cleanups. So my last slide will be is really who is our customer. So when we started the presentation, I am the Director of Sales at FORMULATRIX. So of course my answer is going to be everybody's our customer, but that's a very true statement. So the Mantis itself fits in labs that have very low to medium throughput. We work primarily with customers that, let's say, have a handheld pipetter and they're new to automation, or they've recognized the fact that they'd like to reduce their reagent consumption but can't do so with handheld pipettes. So we're a perfect solution for them. They can, we don't even install our instruments. They come out of the box. A user can plug it in and within 10 minutes be dispensing and running their assays. We also work well with the folks that have every liquid handler under the sun or fully integrated systems. So oftentimes we see, I have a large integrated platform, but I really only want to run 3 or 4 plates today. So then it doesn't make sense to really fire up the platform. You can utilize our system to do semi-automated sample prep and really achieve kind of what you're looking for, but you don't have to spend all that time getting your fully system up and running. So we do sell, and we do have customers that are on both sides of either your low and medium throughput all the way up to your fully automated 1,000 plates a day type atmospheres. So, and I think that is it.
All right.
So any questions you have, you can hold off till the end, and I'd be happy to answer anything for you.
Great, thanks Paul. That was a really nice overview of the Mantis and Tempest and how they can benefit researchers that are performing those genomics applications. So next we have Denise Cruz, the Senior Market Development Manager for Genomics at Labcyte. For over 14 years, Denise Cruz has marketed products and technologies in the life science research and applied markets. She's contributed to the launch of products from DNA sequencing, food and environmental testing, and even forensic DNA analysis. Denise is currently responsible for developing and managing the execution of Labcyte's overall marketing strategy in emerging markets. The floor is yours, Denise.
All right, thanks for the introduction and for the opportunity to provide some information to your attendees on Labcyte and our liquid handler. Let's see if I can get this to work. There we go. So Labcyte, oh dear, sorry about that. Hit it once. There we go. Labcyte invented the use of focused sound energy for liquid transfer, and we began shipping our flagship product, the Echo Liquid Handler, in 2004. Initially, the Echo was used by large pharmaceutical companies for high-throughput screening and sample management, but over time, more areas of research have become dependent on the Echo for high-performance liquid handling. With 58 patents in the US and several global patents, Labcyte is regarded as experts in the use of acoustic liquid handling throughout life science research. Additionally, we have also developed products to complement our Echo liquid handlers and provide our users with comprehensive solutions for their various workflows. We offer a robotic platform to integrate the Echo with other devices that we call the Access Workstation. We have microplates that are pre-qualified for use with the Echo liquid handler to ensure high precision and accuracy. And we also have a suite of software applications that guide our users through the steps that are required to develop a protocol for the Echo. The applications are purpose-specific, and they rely on graphical layouts of source and assay plates so that users can visually see what will happen when. Next, I'm going to show you the different Echo liquid handler models that we offer and give you some information on our technology. We offer multiple versions of the Echo to target different areas of research. Our 525 model transfers fluids in increments of 25 nanoliters, and our 550 and 555 models transfer in increments of 2.5 nanoliters. We initially produced the 550 and 555 systems to target high-throughput screening, where our users wanted to transfer as little compound as possible to conserve their libraries or to avoid having to pre-dilute either samples or reagents. However, as the use of the echo spread to biology labs in other areas, users found that it took too long to reach their target transfer volume with that 2.5 nanoliter increment, and they couldn't reduce their reaction volumes any further. So we developed the 525 model with a 25 nanoliter increment. This is much faster and can quickly build the larger volumes that they need. So I wanted to provide some more detailed information on acoustic liquid handling and our core technology. At the bottom here, you can see that we have a transducer that emits a sound wave to the bottom of a microplate. The sound wave hits the meniscus of the fluid and causes a droplet to eject. Using something that we call dynamic fluid analysis, which I will cover later, we ensure that the power required for that droplet is the same for every drop, even when the fluid changes from either evaporation or hydration. This leads to precise and accurate measurements that are unmatched by other liquid handling technologies. The droplets are produced very quickly at up to 500 drops per second in some cases. The droplets fly upward to an inverted destination plate where the liquid is captured. This sometimes worries people when they hear this, but over the years we've proven that the capillary forces in most microplate wells are strong enough to prevent anything from spilling out. Echoes have been used to transfer a wide range of fluid types and volumes without any issues. A key takeaway here is that the use of our echo liquid handlers not only improve liquid handling performance, but because we don't use tips or make contact with the fluid in any way, there's no risk of cross-contamination or sample carryover. Additionally, since you can go down to low sub-microliter volumes, you can conserve expensive assay reagents and also conserve your precious samples, and significantly lower the cost to produce an assay or screen. And then when you add in the cost of the tips you no longer have to use, and the time for many dilution steps that you can eliminate, and our technology can really enable you to do a lot more with the budget that you have. I mentioned dynamic fluid analysis earlier. With this, echo systems use sound energy to profile the contents of a well in milliseconds before transfer. The information is used to analyze the fluid's properties and adjust the power to ensure a precise and accurate drop volume. For our users, this means that if they have plates containing various concentrations of a fluid, or their fluid properties are changing over time, the echo will compensate and still transfer with high precision. There's no need to create a number of calibrations for the instrument or transfer techniques or anything like that. The echo simply senses and adjusts. As I mentioned earlier, initially the echo was used by large pharmaceutical companies for high-throughput screening and sample management. But over time, more areas of research have taken advantage of the benefits of the echo liquid handlers. In addition to qPCR assay setup, the echo can be used in a wide variety of genomics research applications. This slide shows the impact the echo has had on applications in genomics research. You can see that in every case, the echo can reduce the reaction volumes to save costs, and in most cases, time is saved as well. Add in the reduction of errors and the ability to set up more complex reactions, and the echo can enable labs to do a lot more than what would be practical with other technologies. In the case of qPCR assays, using the echo liquid handler to miniaturize assay volumes reduces reagent usage and costs and reduces assay turnaround time, both while providing greater assay efficiency and flexibility. The risk of cross-contamination is eliminated, and there's no need to pre-dilute samples or PCR reagents, even in reaction volumes as low as 250 nanoliters. Here you can see the impact the echo has on qPCR assay costs. With the reductions in the primers, probes, and master mix used, we see a much lower cost per reaction, which goes even lower when you account for the savings you get from eliminating the tips. So to recap, echo liquid handlers provide several advantages that can help you miniaturize your assays, saving you time and money, and achieving the accuracy and flexibility needed for a reliable and scalable liquid handling solution. That's all I had, and so I'd like to hand this over to our next speaker.
Thanks, Denise. That's a really nice overview of the technology and the benefits of the echo. So next, Sripathi from Johns Hopkins Research Institute. Just a quick bio of Sri. He received his PhD from Michigan Tech in Houghton, Michigan in 2013, and he's chosen to continue his research as a postdoctoral fellow in the Donald Zak Lab at the Wilmer Eye Institute. Sri's career goal is to play a key role in the development and translation of stem cell-based therapies from treating macular degeneration and other retinal degenerative diseases, and he'll be telling us a little bit about his exciting research today. Take it away, Sri.
Oh, thank you, Tom. Thanks for your kind introduction. As Paul mentioned about the Mantis and Dennis about the echo, our lab mainly focus on molecular biology of retinal development and mechanisms of various retinal cell injury and death. We routinely differentiate various cell types of retina from induced pluripotent stem cells and embryonic stem cells. And we routinely perform a small molecule drug screening to identify the neuroprotective compounds and which eventually will be beneficial in the clinic. So today, the crux of my presentation is mainly focusing on 2 microliter qPCR reaction from our small molecule drug screen that is we achieved from the stem cell derived retinal pigment epithelial cells, and we pretty much achieved this using the Mantis and liquid echo as a liquid handling system. So just brief overview of the human eye. If you see the pink color back of the eye, this is called retina, and it's a multi-layered tissue connected by optic nerve to the brain. Of course, this, like the normal macula is a central portion of the retina, which is necessary for a sharp vision, like driving a car or reading a book, or even recognizing faces. But unfortunately, this macula undergoes the dedifferentiation and degeneration, which we commonly call macular degeneration. This macular degeneration is irreversible blindness in Western world, and one of the major region is degeneration of this retinal pigment epithelial cells, which resides back of the eye. So we really want to understand the mechanism of the RPE dedifferentiation and dysfunction during the AMD. In Zach's lab, primarily I'm interested in differentiating these retinal pigment epithelial cells from stem cell derived iPS cells and embryonic stem cells. So we have developed a protocol that takes like 3 to 4 months to achieve the fully matured RPE monolayer. In the panel A, you could see here like the dotted cells called RPE are not fully differentiated. After 3 months, we achieve like the fully differentiated RPE monolayer that is in the panel B, and panel C and D. To confirm the RPE nature, we analyze the RPE specific markers, including RPE65 and PML17 by flow cytometric analysis. But during the disease condition, we hypothesized that these epithelial cells undergoes mesenchymal cells morphology, and this is commonly called EMT. EMT is well studied in cancer during the tumor metastasis, but it is very less studied in the retinal degenerative diseases. A lot of studies showed that EMT is a major factor that relates to the RPE dedifferentiation. So we want to understand what are the factors that inducing the EMT during the AMD, and eventually we want to inhibit the process of the EMT. So from taking an advantage of cancer EMT markers, we identified both EMT and RPE markers in stem cell derived RPE cells. To mimic the AMD-like phenotype, we treated the fully matured RPE cells with growth factor TGF-beta and TNF-alpha in iPS-derived RPE monolayers. And we quickly analyzed the different genes that relate to EMT. Those are like SNAIL, NCAD, and smooth muscle actin. They are significantly upregulated in the stem cell RPE. And if you see the RPE65 and dystrophin, those are like the key RPE factors downregulated during EMT. So we really wanted to understand what's the causes of the EMT, and we focusing on the doing the small molecule drug screening. So qPCR screen is like a very cheapest and reliable and faster way to identify the genes that are involved in the EMT. So we use this small molecule high content screening platform. So initially we treated with the kinase inhibitor library that contains 384 drug concentrations. And then after 48 hours, we treated with another dose of small molecules. And then we induced the EMT using the TGF beta and TNF alpha. After 24 hours, we lyse the cells and get the RNA, and then made the cDNA using again the small molecule treated samples. And this cDNA is again the miniaturized using the Mantis and ECHO. The 2 microliter cDNA contains 100 nanoliters of cell lysate and 1.9 microliter of the RT buffer and water. And we achieved this in low dead volume echo plate, which is only 2 to 4 microliter of the dead volume. And then we performed RT conditions. And using this RT, we performed miniaturized 2 microliter reaction. That is like we used only 1 microliter of the so advanced master mix and 800 nanoliter of water. And then we used only 100 nanoliter of cDNA and 20 nanoliter of 100 micromolar primer by echo. And we analyzed the few RPE and EMT markers. And when we are treating with the drug, we don't have to use all these tip-based methods, but all the compounds behave differently. So with the echo, we took a great advantage of the dose response curve. So we created the different doses of the compounds using this software, and then it also does like the complete like the backfilling with your DMS4. And then it calculates EC50 and IC50 and design the plate layout, and in any well format 96, 384, even 1536. And another advantage, if you're using the multiple drug libraries, we can create the barcoding strategy so that you can use the various amounts of the drug with the different libraries. And of course, you don't have to worry about anything, and just you need to change the plates between each library. So it is a great advantage with the reliability and accuracy when you are dispensing the drug. And before we had a Mantis and Echo in our lab, of course, we used to do 20 microliter reactions in 96 well plate and 10 microliter reactions in 384 well plate using the manual pipette. But the disadvantage of this is like, of course, all you know that the master mix is super expensive, and that doing each column or row, it's time consuming and labor intense. And we also found the inaccuracy between even technical replicates. So we really wanted to do a miniaturized reaction to save reagent. And then we started optimizing the qPCR 2 microliter reaction. So here we made a homemade cell lysis buffer, and then we treated each well in 384 well plate with different volumes of buffer, like 4 to 12 volume. And from that, we added those lysate into echo loaded volume plate, and then we dispensed 100 nanoliter to 1 microliter. And we found that like the lowest volume 4 microliter lysis buffer, and from 100 nanoliters of lysate, it giving a great amplification curves and the melt curves. So the greatest advantage of this miniaturized reaction is like we are reducing 5 to 10 folds of the usage of master mix. And of course, it is very faster, reliable, and very accurate. And it is completely tip-free based, and then no labor intensive. And then this is the quick Mantis a screenshot. Here you can see that the 384 well format, which I did in a 384 qPCR reaction. It is as simple as using your Excel spreadsheet. And of course, in the upper right corner, you can see the Excel icon. You can click that Excel icon and you can enter the amount of volume. In this particular experiment, I used 1 microliter of standard master mix and 800 nanoliter of the water, and then dispensed 1.8 microliter total volume. And if you see the Mantis, so here the quick video which I used to dispense for the master mix into 384 well Bio-Rad qPCR plate. So it picks up the chip and it's doing the priming, as Paul mentioned. Yeah, and this is a very fast, like it took less than 12 minutes to dispense entire 384 well plate. And meanwhile, we can set up the echo to dispense the cDNA and primer. Okay. And then for dispensing cDNA and primer, we use the echo plate layout format. And here I'm dispensing my cDNA from 384 echo plate to 384 qPCR plate. Of course, you can do from any well to any format, like 96 to 1536 or 1536 to 384. You can play whatever you want, but in this case, like we have like the 384 qPCR machine from Bio-Rad, and then I had this beautiful reaction setup. And then, sorry, this one is like the rotated, but this is the, oh, okay, it's working great now. So this is the echo. It is as simple as using your traditional video cassette. Now here I loaded the cDNA source plate. And then, yeah, this is the destination platform. Here I'm loading the 384 qPCR plate. Yeah, and as Dennis mentioned, the liquid handling take care of everything. All you need to do is like just loading the plate. And then you can use the 384 PCR from Bio-Rad. And we only optimized, since it is a low volume, 105 the lid temperature, and then all the protocol is same. So the overall dispensing master mix and primer and cDNA takes like less than half hour, and then the PCR reaction is another 1.5 hours. So in total, like 2 hours, like you can get the complete result of the 384 well plate PCR. And if you see some of my CT values from the drug treated samples from the RPE cells, the CT values are so tight, and even between the replicates, and then you can appreciate the melt curves between the samples. And we download the PCR data using the CFX software. You can analyze the data by using delta CT method. If you are having the multiple samples with dealing with the high content screening, I would suggest you to use like the Cubase, although it's a paid software. It works great. Basically, I always use like the more than one reference gene to take the geometric mean and then normalize the samples. And finally, I would like to show some of my data that is validated from the drug-treated samples. So we treated with the TGNF, that is TGF beta and TNF alpha, to induce the EMT. The first, the black colored bar is a control without any treatment. The third one is TGF-beta plus DMS for control. And this one, 48 to 4 micromolar dose response. And again, I did the 2 microliter reaction, like which I shown earlier. These are the RPE65 RPE specific genes, and the bestrophin is another RPE specific, and CRX. If you see the data here, you could see the RPE65 expression is higher when we are inhibiting the EMT using the kinase inhibitor, and same with the bestrophin, and even it is restoration is even great with the another RPE specific marker CRX. So overall, we achieved quite a nice platform for doing the qPCR in 2 microliter reaction. So most of the lab members follow the same protocol, and their results are very consistent. Definitely, this is very helpful for anyone doing the mini-esterase reaction for your screening assays. With this, I really thank FORMULATRIX and Labcyte for providing me this opportunity, and also their excellent support during establishing this protocol in our lab. With this, I would like to take any questions from you.
Thanks, Sri. That was a really wonderful presentation, and it's so exciting for me to see the synergies between the Echo and the Mantis as complementary products for sample prep in your lab, especially in the context of an interesting high-content screen. So let's see here if we have any questions from. Looks like we have a few. So, what is the accuracy of the well survey for aqueous media?
Well survey and between the wells.
If someone would like to clarify the accuracy of the well survey for AQ media. Okay, we'll wait to see if someone clarifies. So the next question here is, have you ever seen any contamination in this workflow when using the Mantis or the Echo?
No, not at all. As we mentioned, Paul and Dennis is completely tip-free, so we don't see any contamination even within the 1536 well plate. So accurate, yeah, no, not at all.
Great. I have another question here. What types of liquids can the Echo transfer, and does it require calibrating?
Thanks for that. No, we can transfer a wide variety of fluids. We started with DMSO-based fluids when we did drug discovery work, and now we do aqueous fluids up to about 40% glycerols, and it doesn't require any additional calibrations. It handles a wide variety pretty easily. And you can put multiple fluids in one plate. The dynamic fluid analysis allows for that.
Great. It looks like I have another question for you here. Denise, can you use any type of plate with the Echo?
The source plates need to be our plates because they're super flat to allow for the acoustic energy to go through correctly. But the destination plate can be anything that meets the SBS standards that the gripper can hold. So a 96 well, 384, 1536, whatever it is that the lab needs to use.
Okay. Great, and it looks like we have one final question. Can the Echo transfer beads or cells?
It's really dependent on the size of the cell or the size of the bead and how well they're settling during the transfer. They need to be pretty buoyant in that fluid. But having said that, our users have been successfully able to transfer both cells and beads, and the acoustic energy is very, very it's very light, so fragile cells aren't a problem. We don't do any damage to the cells.
Okay, great. Looks like I have another question for you here. Denise, has the Tempest been integrated with the Access system?
I am not sure about that. I would have to get back to that person and check with our Access product manager. So if they wanted to send me their information, or maybe Paul knows.
Yeah, I believe it's on the list for this year. We have a mutual customer that's looking to drive the integration, so we have we're speaking right now, and it looks like something we'll be able to offer this year.
Great, thanks.
Sure.
Wonderful. Okay. So if there aren't any further questions here, I'd like to thank all of our speakers today for doing such an excellent job in preparing for these presentations. I think this has been a really educational and great event. And I'd like to thank all of the researchers out there for taking your time out of your busy schedules to be here and learn about not only Sri's interesting research, but also the technology and the benefits of the Echo and the Mantis. So thanks everyone, and goodbye.
Thank you.
Thank you.
Thanks.